We are here today to talk about VEuPathDB and what's new in Release 59. I'm Suzanne Warrenfeldt, and I'm joined by my colleagues Evelina Bacinco and Gloria Geraldo Calderon. And we will be talking about Release 59 and demoing and demonstrating some data there. Release 59 was kind of heavy on features. We got quite a few new features across all of our sites. Single cell RNA-Seq is now new in a beta version, and data went to 3 different sites: PlasmaDB, TriTripDB, and PurePlasmaDB. So I would like to talk about the features for a little bit. For that, I will go to VEuPathDB on the home page. And go to the news, right? In VEuPathDB, which has data that accesses all of our sites, we can go to the news and then scroll down and look at news from any of the sites. Oh, I forgot to mention that we are here to also answer questions as we're going along. So in your GoToMeeting controls, you should have a questions panel. If you type a question in there, we can answer it straight away. So I'm going to scroll down to the TriTripDB news release information and talk about the features there, or reference that as the features. So there are 4 features. We have Apollo data is now accessible from our site search. So I'll open a new VEuPathDB. If I use the site search to look for, say, genes with the word kinase in their annotations, and then I filter on genes, I see we have this new category, Apollo annotations, and I can look at genes that have the word kinase attached to them in new annotations added from Apollo. Apollo, of course, is our community annotation platform, and users who notice incorrect gene models or have new functional annotations can use this platform to add that information to the gene. And that is what you're seeing here. There are 20 genes across all of our sites that have new information attached to them that include the word kinase. So if you're an annotation guru, there's a new tool for you here. Let's go back. So our gene page navigation has some improvements. Let me go back to our kinase. These are our kinase results we were looking at earlier, and I'm going to click on a toxo gene, go to that gene page, and look at the transcriptomics section. I'm going to use this thumbnail here to go down to the transcriptomics section. And I see that it actually has, 3 sections: a summary graph, a table called transcript expression, and the user data set transcriptomics. The summary graph, of course, is a graph that has that graphs every experiment, the data from every experiment, and the transcript expression table has each row is the details for each experiment. Well, the navigation improvement that we have in Release 59 is that the transcript summary graph actually links now to the transcriptomics table. So for this gene, I'm looking at the transcript summary graph, and I see that, wow, this experiment is really kind of interesting. There's a wide range of expression for this gene, so I want to look at its details. And so if I hover over the title of this experiment and click on it, it'll take me down to the transcript expression table and open up the details for that experiment. So an improved way to navigate the expression data on gene pages. I'll go back to VEuPathDB, and we have a new data and a new data type, more or less, in our VEuPathDB Release 59, and that is single-cell Rna-Seq data, and we've integrated that into a beta version. We might want to make changes in the way that we represent this. It's not 100% finished with development, so we're calling it a beta version. The 3 sites that received this data are PlasmoDB, TriTripDB, and PurePlasmaDB. So what we have is a search that returns all genes that have expression in the in cells for that experiment. So let me go to, I'll just go to PlasmoDB, and I'll say searches, and look for single cell RNA-seq, and I'll go to that search. We have 1 experiment down here. If you look at the description and the data sets used for this search, we have 1 new data set that is single cell RNA-seq, and we just have a very simple search. It returns all of the genes that had expression in cells from this experiment, right? So I find that I have 248 cells, which might seem low if you are familiar with single-cell RNA-seq, but this, the data that we received, these are very high confidence genes from this provider. So 248 genes with single-cell data. And then we offer the Cell by X, the Cell by Gene platform for exploring the visualization of expression between samples and genes. And so let's just go and look at that. I'm going to use the navigation to go to the transcriptomics section. And look at the single cell RNA-seq. We have a table just with a little bit of summary information, but if I open that up, then I'm able to, it will load the Cell by Gene Gateway, which uses UMAP to display the information. Sorry, it's taking so long. And then we also have links to explore the full data set. And oh, here we are. So on the left here is a visualization of all of the cells within this experiment that have been clustered. And on the right is a description, a graph of the expression for this gene. We also offer this a link out to the Cell by Gene platform where you can investigate all of the clusters. You can color genes to help you help with a little bit more visualization. You can choose different gene set clusters, and we have linked to a YouTube video and a tutorial for help in this matter. Okay, the last feature that all of our sites got, more or less, is the AlphaFold protein structure database information. Let me see. Yes, how AlphaFold data is now available, and AlphaFold, of course, contains structure predictions for a wide range of organisms. We have associated, so we downloaded their database, version 3 actually, and associated their predictions with genes on any of our sites via the enzyme commission numbers. So not every gene has a structure in our database, but we created a search. you know, we created a search for AlphaFold. So if I go to search and look for alpha, this will return all genes. I'm going to choose all organisms here. So you can see that although we have 646 organisms in our system, we have 318 of them actually have genes with AlphaFold predictions. And we're going to find out how many genes that means here in a second. So there's 147,000 genes on our sites that have AlphaFold structure predictions. And let's just go to I'm going to only look at Acanthamoeba castellani genes here. There are 15 genes in that whole genome that had predictions. So let's go to one of those and see what we've done on the gene page. So here I am at that gene page. I'll open up the content section and look for AlphaFold and go to the structure analysis section, which has 4 tables, 4 bits of data in it, and 2 of them are new. One of them is called AlphaFold. Try to make that bigger, which has a table with data about the predictions. Some genes have many more predictions than this, and then we choose one of those genes to show the visualization down here in this 4th bit. And this is, you know, really very useful if you're interested in structure predictions and you're really studying this this gene. Its protein product structure is here. You can hover over these any acid here, and it'll show you like that's turning red, you know, down in the and vice versa. You can hover over the structure down here and see a red highlight in the sequence above. And then there's other, you know, things that you can do that. If you're interested in this, you can learn more at the AlphaFold site on how to use their visualization. One slight caveat is that as we were developing this, AlphaFold released their release 4, which has many, many, many more predictions. I think version 3, which we have integrated, contained like, I don't know, 25 or 30,000 predictions, whereas the new version 4 has like 2 million. So in a future release very soon, we will be updating this information, and so this search here will return many more than 150,000 genes. We'll have many more predictions in our site. Okay, so that is those are the new features. It doesn't look like there's any questions, so I'll go on to talking a little bit about the new data in our sites. And this is just an overview to let you know, if any, you can go back and look at your favorite site and see what new data is there. All in all, we integrated about 89 new data sets in release 59, and here's the distribution across all of the sites. You'll notice that some databases don't seem to get very much data, whereas, fungi might look like it's overrepresented by attention from our developers, but that's not necessarily true because fungi covers many, many more organisms than most of these other databases do. Like Plasmodium is one, the Plasmodium part clade of the taxonomic tree, whereas fungi, is an entire kingdom. So there's lots of new genomes and wonderful data include for the fungi. So, and all of that data spanned many different data types. So AmoebaDB received many new genome assemblies. They were updated to the INSDC records, received 6 or 7 RNA-seq data sets, and then some of those RNA-seq data sets were used by the data providers to produce transcription start sites, which we integrated as gbrowse tracks. CryptoDB actually received a new genome assembly update. HostDB, of course, Fungi received new genomes, whole genome sequencing that we use to determine SNPs, RNA-seq and ChIP-seq. Microsporidia actually received 11 new annotated genomes, and then on down the line. So I will go ahead and demo one thing in AmoebaDB. I'm sure the people who study Amoeba are kind of excited that we have so much new data. So let's look at that. I'll go to AmoebaDB. Okay, so from that table, I saw that there were, well, let's just look at the release here. So the following genomes were updated to their INSDC versions. So we have this partnership now with NCBI, and we're trying to have to reduce the variability between genome versions among all of the databases. It's just sort of stuff that goes on in the background. And so, we do not own these genomes, the data for these organisms. It is the people who collect this data deposit it in repositories, and that is the official location where this data is saved. And so some of these genomes and annotation we had actually received from data providers before they were put into the INSDC version. So we just updated these to make sure that we have exactly the same assembly as the INSDC. And look at all of this RNA-seq data that we got, that we were able to integrate. It's very recent from 2020 or 2022. So I just like to show you one of those searches to remind you how to do it. So if I go to searches and the RNA-seq evidence track search page, I get a list of all of the RNA-seq data that is integrated into AmoebaDB. And if I just search for the numbers 59, I'll see everything that went into release 59. And let's just do a search. I'm going to choose this data set called Xenic, normal, attenuated and virulent, colon cultured E. histolytica. And I chose the differential expression search. We were able to calculate statistics for these expression values because of the experimental design. There were replicates for each condition in the experiment. And so I will choose to look at this experiment in the sense direction, comparing virulent colon to the culture of virulent cells, looking for genes that are up or down regulated, let's say the fold difference of 2 and a p-value of 0.01. Once I have all my parameters correct, I'll look at get answer. And I see that I have almost 1,200 genes returned by the search. And so let's go to one of those gene record pages. And I'll use this to go to the transcriptomics section. And how about our new handy dandy tool that we have? I find my experiment that I'm interested in looking at, exenic of colon versus cultured, and I can click on that title and go to the details here. Just as a reminder, then I can go back to my strategy and say, well, it's awesome to know that these are overrepresented in colon versus culture. But I would like to know if they're secreted. And so I'll intersect that with another search for signal peptides, which we predict for every genome that's coming in into our sites. I'll say collect all, select all, and run that search. And I'll find that 122 of them have higher expression in when they're, not cultured, and they also have signal peptides. Okay, so I will stop sharing my screen, and I will pass the baton on to Gloria, who can talk about vector-based data in Release 59.
Thank you, Suzanne. Are you able to see my screen?
Yes, that's
okay.
great. Looks great.
Thank you. Excellent. Hello, everybody. So as Suzanne just mentioned, I am going to tell you what we have new for vector-based in this release. About the data, we have this genome, Culex kinkephasiatus Johannesburg 2020, was released before, but now serves as the reference species. So let's go to data, organisms, genome information, and statistics. And here we have 60 genomes. Type Culex. It's not necessary to type the whole name. So you will notice now in this table that it says is reference. So now this one becomes the reference. And what this means is that now this is the species that is also available for annotation in Apollo. There are a few exceptions in which reference, there is some species that are not referenced that are also available for community annotation, but most of the times is the reference strain. And this represents a nice improvement in the assembly and gene set prediction for the community. So if you have any feedback or questions about how or why we changed the reference strain, please let us know. We have a new genome. It's Triatoma infestans. That's a kissing bug. Triatoma. So the main difference with this new genome that we have is this one has no gene set prediction, unlike the other one. So I'm going to show you, if I click here, I go to the gene page, and I'm going to show you what that means in terms of the resources that we have available for you for that genome. So on the one that we were looking before, Culex kinkephasiatus, if I come to the page and scroll here, so I explore this data set within the website. So I can go to the Genome Browser, I can look at genomic sequences, and I can run searches based on genes. And if you click on any of these links, it will take you to the specific search. Here with Triatoma, we have more limited resources because we are limited to only genomic sequences because there is no genes. But still, we believe this is a very important species to have, in addition to the other kissing bug that we have, that is Rodnius. So hope you can make the most of this species. So we also have this species, Ixodes capillaris. And this is also, let me show you, Ixodes. Okay, so we previously had this cell line, Ixodes capillaris ISE6, and we have got the YCOL reference genome assembly. And notice the difference here with the supercontigs. So notice that YCOL strain has more than 300,000 supercontigs, and the new reference assembly, which is in second row, it has 648. So this is definitely a huge improvement for the community. What we are going to do in a future release is that now VEuPath strain will not be anymore the reference strain, will be this new PAL Lab Hi-Fi strain, and that will be the one that will become available for Apollo. And that means also that all the RNA-seq data that we have, I think it's not loaded here, RNA-seq data, yes. So all the RNA-seq data will move from VEuPath strain to the new strain. And as I just showed you before, if you click on that name of the species, you will come to the gene record page, and you'll see when was released, and here all the links for the data that you can explore with this new genome. Okay, about the Rna-Seq experiments, we have one paper with two different experiments, one with data for Aedes aegypti and another experiment for Anopheles gambiae. And this is a body atlas with many, many different tissues. So for Aedes aegypti, they have all these different samples, how body, head, thorax, abdomen, gut, many different time points. And here we have details about how the data was collected, but for more details, here's the link for the paper and the different searches that you can run with this data set. From the same paper, they also analyze Anopheles gambiae with a few less samples, but still really nice data atlas to analyze with RNA-seq. And another experiment that we had, so this is also from a paper that got two experiments, but the other experiment got released in a previous release. And here the name is Tang. So if I go to the page and you want to see what else do we have for the same author. So again, you come to the page here, but something else that you can do is you come here to the home page, you look for the RNA-seq data. And as Susan was just showing, there's different ways to search here. I can say Tang, and this is the one that I just showed you, but from the same author, we also have this data set. And you can scroll to the bottom of the page and come to that same record page that I was just showing you with a diagram of, the experimental design for this new data that we are including. And in addition to these big RNA-seq studies, we also have 3 new population data sets for the MapView. And that's all I have to show you. If there are not any more questions, I will pass it along to Eve.
All right. Thank you, Gloria.
Sure.
I will make myself a presenter. All right, you should be able to see my my window now.
Yeah, it looks great. Yeah.
All right, sounds good. All right, so I will briefly walk you through the data sets that we have for fungi in this release. And as Suzanne has mentioned, we have genome sequence and annotation, SNPs data sets, transcriptomics, and also ChIP-seq. When it comes to genomes, we have loaded quite a few of various pathogens, whether this is plant pathogen, for example, Alternaria alternata, that affects over 300 different host species of plants. We also have Pythium, which is an oomycete that could be a human pathogen in certain circumstances. We have Cryptococcus. We also have Batrachochytrium, which is a pathogenic chytrid fungus that infects an amphibian species. And we have other interesting genomes, including those species that are parasitic on insects. Following that, oh, by the way, I did want to mention that if you're ever interested in learning about these sort of the specifics about the dataset, and it has been shown before by Gloria, all you need to do is navigate to the dataset record page. And on this page, we'll give you a few more options in terms of how the genome can be searched, in case you haven't seen this before. So this is these are all the searches available for this particular genome, including gene-specific searches. And this means that we have put the records for the genome sequence and other data through our pipelines that help you not only to identify genes, but also cross-reference this data with various predicted signatures predicted domain signatures. And you can also interrogate this data using the genomic segments or genomic sequences searches that are available as well. And obviously at the very bottom, we also want to make sure that we list information where it's coming from, such as in CBI by a project, and we link out to the page where we downloaded the dataset from. Right, coming back to the datasets, now I just want to quickly mention the whole genome sequencing datasets that we have. In this particular release, we have data for human pathogens such as Aspergillus fumigatus, Histoplasma capsulatum. We also have pneumocystis, and pneumocystis isolates have been, I believe, yes, also collected from the clinical conditions. And we also have two data sets for agriculturally important plant pathogens such as Paricularia ariziae and Zamaseptoria tritici. So in terms of the investigation of the whole genome sequencing data, let's take a quick look at the SNPs that were generated for the whole genome sequencing data collected in Canada. This was a large study that have looked specifically at the histoplasma isolates. So histoplasma is an interesting pathogenic fungus that have been shown to spread now to various regions within the US and also Canada, possibly due to the climate change or disturbance of soil. And this particular data set can be examined by the listed searches as shown right here. And for example, you can analyze your data with a search for a specific SNP characteristics. So let me navigate to histoplasma really quickly. And G217B is a reference genome that we have used for this sample. I can click on the data set, and here's the data set collected from Canada. And let's say if you're interested in exporting all SNPs, you can do so by using the sort of default parameter. But if you want to quickly navigate and look for non-synonymous SNPs only, you can do that as well. So the only little bit of sort of tip that if you are looking for any of the subcategories of all SNPs, in order to return the sort of the subcategory, you must select at least one or more SNPs to return searches. Otherwise, you will be returning all SNPs within the dataset. But anyways, this is how you can quickly sort of run the search. And once you click on the get answer, you'll come back to a search strategy as Suzanne have shown her on her screen in the past, where you'll essentially see the your results, and you can expand this strategy further. Now, just quickly coming back again to the news section, I will go back to the release again. Release news, and scroll down to the transcriptomics. So we have quite a few of transcriptomics data sets. And again, these cover various pathogens, model organisms such as Aspergillus nigricans, and that also includes a number of data sets for Cryptococcus neoformance. So you can assay these resources just as you would before. The RNA-seq data can sort of come in different flavors in terms of the usage of the biological replicates. So if your data set, for example, does have biological replicates, you will have a differential expression option to search this data set. The differential expression pipeline requires at least 2 or more biological replicates to be run on the data set. So, and we also now provide a specific data set if your data is stranded, such as you could look at the transcripts upregulated on sense or anti-sense data when this information is available. Now, I also want to quickly just talk about the Cryptococcus neoformance data. There are a series of data sets available from the paper published in 2020. And the authors in this paper have specifically looked and experimentally determined the beginning and the end of both coding regions and also transcripts in 2 Cryptococcus species, KN99, a lab isolate, and also JAC21, a standard reference genome for Cryptococcus neoformans. And so we now have this data available, and this data is particularly useful, especially if you're working with this pathogen and is interested in looking, let's say, at the transcripts, the boundaries for each of the transcripts. And so this data can be mined through a standard Rna-Seq approach. However, you can also look at this data in the JBrowse. And so notice that I'm now showing JBrowse, and I navigated to JBrowse by going to the Tools, Genome Browser, then selecting my genome from the drop-down menu and activating the tracks that I was interested in. And so here you could look sort of for a specific gene. Here's a multi-exon gene and the specific expression in different conditions and across also different strain. You can look at this sort of information within the specific region, or you can also look at the larger regions if you're interested in neighboring genes. Now, coming back to to the other types of data that we have ChIP-seq data, and in this case, there are 2 data sets for Aspergillus nigellans, where people have looked at the modifications occurring during canidia and canidiation, and also in spores, which has important implications for development in Aspergillus nigellans. All right, and with this, unless there are any questions, I will turn this back to Suzanne.
Yeah, I don't see any questions. It's one more thing, since all of your transcription start site data, you know, is the same thing as what we had shown in, or what we put into AmoebaDB. I thought that I could show a couple of the AmoebaDB tracks. So I'll go to the news first and scroll down and see that we integrated these transcription start site JBrowse tracks for 4 different organisms, and Amoeba dispar, Histolytica, invadens, and Moshkovsky. Sorry for my terrible pronunciation. All right, so we can view all of these in JBrowse. As Eve mentioned, we have analyzed the Rna-Seq data, but also have JBrowse tracks in AmoebaDB for these for the results of that Rna-Seq when the researcher actually looked for the transcription start sites. All right, so I'm going to Tools and Genome Browser. And the Genome Browser, of course, will focus on one genome at a time, and I end up at E. histolytica. So, and I can see that when I'm in Genome, I'm in Amoeba histolytica. So if I want to see the transcription start site tracks that we just added, I would go to the Select Tracks page and I can search for that. I'll type in TSS for Transcription Start Site and then go ahead and turn those on. And then my track appears down here, and what I see is I don't necessarily have any. I will scroll out. Oh, this is a very small config. So I'm going to go up and choose, hopefully, a much larger one, and then see transcription start sites for each of those. So I'll zoom into what I'm seeing here is the annotation on top, community annotations from Apollo, the next track, this track for evidence for introns. So as we analyze all of the RNA sequence data, then we create these tracks that talk about, that show data for introns that match the annotation and introns that possibly exist in the biological organism but don't necessarily match what's in the annotation. Okay, so I'm going to turn those off so that we can see our transcription start site at the bottom. So I will look, drag into zoom in on an area, and I can look at each of these transcription start sites. I'm going to click on one and get the information to see exactly where it is. So this is really useful. If you are studying a pathway that has this certain gene, you can come and look here and make sure that transcription start sites actually exist where you think they do. And again, we have this data for several different organisms, E. dispar, E. invadens, histolytica, and one other. Okay. So it is 10:43, and we've given an overview of the data and demoed some of it, that it's in release 59. If there are no questions, I think we could wrap up a little bit early. Okay, silence. Sounds great. And I hope everyone has a wonderful day and they find our release 59 useful.
Bye, everyone.
Bye-bye. Take care.
Bye.